time. And we're now going to have an extensive session on in vitro cell culture models, advanced in vitro models of the human lung, of the GI tract, of liver for hazard characterization. And this is going to be run by Kirsty Meldrum and her team. The presenters also extending to Barbara Drasler, Rui-Wen, Angela Kämpfer, Samantha Victoria Llewellyn, and Marije Niemeijer from the Petrels project. I guess Kirsty is going to start, and I will shortly upload or launch her film, which she provided, and she will probably introduce the entire team herself. So I give the floor to Kirsty. Please start with your presentations, and I will try to assist you as best as I can by uploading presentations. We can, however, not give a microphone to each of you at the same time. Otherwise, we're going to exceed the number of possible talkers at the same time. So, Kirsty, you may unmute your microphone.
Yes, hiya.
To introduce your session, please.
So yes, thank you for that introduction. So our session is all about the different models we've developed within patrols. So we'll have a section on lung models, liver models, and GI tract models. And to start off is my presentation on the lung models that we've developed in Swansea.
Thank you very much to the organisers of the conference for allowing patrols to present the work we've completed on developing new advanced tools for nanosafety testing. We appreciate being invited to discuss the new models we've made. My name's Kirsty Meldrum, and I'm going to be presenting on behalf of Swansea University and the models we've made with the lung, specifically on developing an advanced in vitro alveolar human lung model. So the lung is the primary exposure route for nanomaterials, especially for occupational exposures. It's the important organ for respiration, and this is what gets oxygen into our blood. There's three main areas of the lung: the upper, the middle, and the lower. And these three different areas are made up of various different cells, which is what we've then focused our models on within patrols. An additional factor to consider when deciding what region of the lungs to study is what size of particle you're interested in, and therefore the area within the lung it will deposit. The smaller the particle's diameter, the lower down the lung it has the potential to deposit, and therefore the lower down the lung you should focus your work on. So this diagram here indicates the different sizes of particles and the different regions of the lung that they would potentially deposit within. So the top is the pharyngeal and the upper airway. The green is the bronchial area and the middle of the airways, and the red is the alveolar unit that the smallest particles would deposit within. The area of the lung that we've decided to focus on within Swansea University is the alveolar region, and this is the region that gas exchange occurs within. This region is made up of type 1 epithelial cells and type 2 epithelial cells. The type 1 epithelial cells cover around 95% of the internal surface of the lung and are there for gas exchange, while the type 2 epithelial cells are the secreting cells. There are also alveolar specific macrophages present within the area. University, we had three different cell types that we wanted to focus on to make our alveolar cell model. These were A549 cells, small airway epithelial cells, which were primary cells and not a cell line, and NCI H441 cells. These are all type 2 epithelial cells. So the first thing we wanted to do was to culture and characterize these cells. So we cultured these for a total of 14 days. These were grown both in submerged cultures and air liquid interface cultures. So initially we grew them in submerged cultures for 7 days, before then switching them to an air liquid interface for 7 days, which is where we took the apical media off of our transwell inserts and just left the basal media. We then characterized these models, and we looked at cell viability and cell count, the membrane integrity, and this was done using TEER and blue dextran, and looking at various cytokine and chemokine releases, specifically IL-6 and IL-8. This led us to deciding what cell type we would then carry forward. We did not get on with our small area epithelial cells. We couldn't get them to grow in our transwell inserts, and this was a massive hurdle for us. But also, our NCI H441 cells have an embargo on them, which means they cannot be used in industrial settings. So therefore, within this Patrols project, we took the A549 cells forward onto our next steps of nanomaterial testing. So this looks like a lot of data. However, the key take-home message from this data is that when you build a model, you need to understand over time how they grow and how they behave. It's important to understand when the optimal time would be to switch them to an airlift interface, as well as when your exposures should be completed. So with our A549s, we decided that it was 3 days submerged culture growth and 1 day at an airlift interface before we would expose them to our nanomaterials. And this region is highlighted with these red boxes within our graphs. And this would just give us an indication of when was the most optimal time our model was the happiest for us to complete our exposures. So building the model. This is a quick overview of what our end A549 and macrophage model looks like as a cartoon. So we have our A549 cells in the blue and our macrophages in the purple. And I note that all of our components are pre-warmed to our 37 degrees before we're using them. So this is one we made earlier, and this is what we're aiming for our model to look like. Our A549 cells come from ATCC, and we grow them in RPMI media, which is supplemented with penicillin streptomycin, 10% FBS, and 1% L-glutamine. We use inserts from Falcon as well as companion plates also from Falcon, and we use a mixture of 12-well and 6-well inserts depending on the endpoints we want to look at and focus on within this project. So initially, we want to seed our A549 cells alone into the transwell inserts with media both on the apical and the basal side, which is indicated by the red on this diagram. So we start by taking our transwell inserts out of their sterile packaging and placing media into the basal side. Now the volume of media you place into the basal side depends on the insert size you're using. For a 6-well insert, it's 3 mils. For a 12-well insert, it's 1.5 mils. We then take the transwell inserts using tweezers and being careful not to touch the membrane with any fingers or any other objects within the hood and place them into the wells. We then ensure that our membranes are within the grooves within the plates and then place these into the incubators ready for us to seed onto them. So the next step in seeding and building our model is to trypsinize our A549 cells off of the flasks they've previously been growing and then seed them onto our transwell inserts. These flasks are washed 3 times with PBS. For a T75, we tend to use 10 mils of PBS per flask. And we wash this to ensure there's no media residue left within the flask so the trypsin doesn't work. We then take 5 mils of our trypsin, which again is lab specific, and there are other laboratories that use a smaller volume, and place it within our flask. This is then placed into the incubator and left to incubate for 5 minutes, taking a constant looking at the cells to ensure that they've been trypsinized properly, and then taken back to the hood. So now that our cells have been incubating for 5 minutes in the incubator with the trypsin on, we now need to neutralize it. So all the cells have detached from the bottom of the flask. We neutralize the trypsin by adding 10 mils of FBS media into our flask and swirling around to ensure that all the trypsin has been neutralized. We then pipette this into a centrifuge tube and take it to be spun. Once we've obtained our cell pellet, we then pour off our waste media and resuspend our cell pellet in a mill of media. This is done gently as we don't want to disrupt the cells or kill any of the excess cells. We then calculate how many cells we need for our seeding, and we dilute our cell pellet as required. Then, taking the plate from the incubator, we seed 500 microliters of the cell suspension onto each apical side of the membrane, if it's a 12-well plate, and 1.5 mils of this if it's a 6-well plate. The next step of our model is to prepare our macrophages. There are other macrophages that you can use, as well as different differentiation methods. However, within Swansea, we use THP1 cells and we differentiate them using a PMA method. This differentiated method has been characterised within the university and within the group. As I've said previously, it's important to know what you're working with and what the cells are like once they've been differentiated. You need to understand the cells before adding them into a pre-characterised model. So we seed our cells at 1 × 10⁵ cells per mil, and we add 20 nanomolars of PMA for 48 hours. We then change the media to fresh media, and we allow this media to be on for 24 hours before removing the macrophages from the flasks, seeding them onto the transwell inserts, and having them in air liquid interface for 24 hours before doing our exposures. So 4 days after we have seeded our A549s onto our transwell inserts, they're then ready to be switched to an air liquid interface. So we remove our apical media and we change our basal media, so the media on the bottom is fresh. 24 hours after switching our differentiated THP1 cells to media, we then repeat our washing steps as we've seen previously with our A549s. So we wash our THP1s 3 times with PBS before adding 5 mils of Accutase onto this. Now, we use Accutase instead of trypsin as it's a gentler method and it works in a different way. We incubate this for 10 minutes within our incubator, and once it's been incubated and the majority of the macrophages have detached from the flask, we neutralise our Accutase using 10 mils of our cell culture media. This is double the volume of the Accutase we would have placed into the flask. We resuspend we centrifuge to obtain a pellet. We then resuspend and count our pellet, and we dilute our cells to the 1 × 10⁵ cells per mil we require for our transwell inserts. On a 6-well insert, we pipette 500 microliters of 1 × 10⁵ cell suspension onto the apical side of the membrane. Once we've pipetted this 500 microliters onto the apical side of the transwell membrane, we then allow 2 hours within the incubator for the THP1 macrophage-like cells to adhere to the A549 cells. After this 2 hours, we then remove our apical media and the model is switched to an air liquid interface, so there's only media on the basal side. This model is then allowed 24 hours to accumulate and acclimatise to the air liquid interface conditions before using for any exposures, be that chemical or nanoparticle. The bottom 2 images on this slide actually show a macrophage on our epithelial cell layer. So from the cartoons above, we can actually then see a physical representation of the macrophages on the cell layer. So the right-hand side is a TEM image, so a transition electron microscopy image, whereas the left-hand side was done with our confocal microscope and fluorescent staining. So we can see that these macrophages actually adhere to our epithelial cells as we'd want. Now that we have our model and we've allowed it to acclimatise to the air liquid interface for 24 hours, there are various exposure methods that we can implement. The traditional and most commonly used exposure method is a submerged exposure. This is where the whole of the apical side of the membrane is filled with media with our chemical or particle of interest and exposed to the cells that way. The next method, which is slightly more physiologically relevant, is the quasi-alley exposure. This is where a specific volume of media is placed onto the apical surface of the membrane, but does not cover the whole surface, and therefore is quasi-exposed to the membrane. The next 2 exposure methods are more advanced and again try to replicate more of a physiological exposure method. This is both the dry powder exposure and the liquid aerosol exposure. These have now both been obtained by Swansea University, and we look forward to actually implementing them within our work. So this is an example of one of our exposure methods, which is the VitroCell Cloud 12. I've placed a QR code on the left-hand side in case anyone would like to have a look at more of the details on the website. So using this system, we place a specific volume of our nanomaterials in suspension into the nebulizer, which is then nebulized into our exposure chamber and onto our transwell inserts that are placed in the bottom of the chamber. We can also then measure the deposition of these particles using a quartz microbalance. This means that we can replicate these conditions quite consistently and are also doing this as part of the Patrols project within various different labs, comparing the deposition we get of specific nanomaterial using this system. So as you can see within Swansea, we focused on the 8549 and differentiates THP1 cell alveolar models. We have focused initially on the quasi-alley exposure method, but are now starting to use our Cloud 12 aerosolized exposure chamber. So thank you very much for listening, and I'd like to invite any questions.
So I do see there's a few questions in the box. The first one about the PMA differentiation. Yes, there are quite a few different protocols for differentiation of the THP1s. And as I said in my presentation, the reason we went with the protocol we did is because it's being characterized in-house. We characterized a range of differentiation periods as well as the concentrations of PMA. And this is what led us to decide to choose our 48 hours with PMA and 48 hours without. For another project we're doing within Patrols, we're actually using a 5-day differentiation protocol for the THP1s with a 3-day recovery phase. So we are using different exposure methods within Patrols, but for this model, it's a 48-hour PMA exposure and then a 48-hour recovery period. On the basolateral side, no, not within this project. We have been looking for seeding endothelial cells onto the basal side of the transwell, but we've only been focusing on the apical side just now. And the pore size we use is the 3 micron pores, but for other cells, they'll need the 0.4 micrometer pores. No, we've not used the machine for printing our compounds on top of the cellular layer, but I think I've seen publications that have done so. So I bet it is fantastic. I would quite like to have a shot with it. So with our primary macrophages, we have got other people within the lab that have been using our primary derived macrophages from blood. Within Patrols, we decided to stick with our cell line just purely for reproducibility and ease, especially now that we are struggling to get hold of blood, with people being a little bit more cautious about donating blood and getting access into hospitals. So we've just stuck with our cell lines, but I know other people within the lab have used the macrophages derived from monocytes, and within Patrols, it's also been used within other partners. So that ratio is based on a previously published literature as well as historical data that have been produced by different labs. The ratio. Yes, exactly. So Barbara is going to show the primary monocyte work. So with the A549s, we actually struggled to get a tier value that would replicate what we read in literature. The tier value we got was maybe a factor of 10 lower than what we read in the literature. But this was actually consistent between ourselves and another Patrols partner. So we weren't sure if in the literature it's reported differently, if it is, say, centimeters squared, or if it's just a tier value. But we struggled to get quite high tier values with our A549s. With our H441s, we did get higher tier values. However, due to the embargo on these cells, we didn't want to take them any further forward. The protocol is being put together now. We're also going to be publishing a paper on this protocol, which should hopefully be with the publishers soon. That's a very good question. Also, one of our other Patrols partners has been looking at the Halby cells instead of the A549 cells. Not within Swansea, we focus purely on the A549 cells.
So I guess this statement of Tommaso belongs to the printer.
Yes.
Question, I guess. Okay, thanks a lot, Kirsty. I guess we are going over to Barbara. Barbara's talk on or presentation I will upload shortly.
Sure.
On inflamed alveolar epithelial model for nanomaterial hazard assessment. Thanks
All right. Okay, thank you very much. Sorry.
Thanks a lot for coming. The stage is yours.
Thank you. All right. So thank you, Martin, for the presentation, and to you, Kirsty, for giving the introduction on the lungs and its compartments. So I will also present an alveolar model. It's a bit a different one than Kirsty presented, and I will present how to prepare a diseased inflamed model in vitro. I come from the University of Fribourg in Switzerland, and we are also part of the Patrols project. So as I said, I will focus on the innermost, the deepest regions of the lungs, and these are alveoli. And as Kirsty said, this is where the gas exchange occurs. That's why this is a very important region. We can mimic this alveolar model again by using these transfer membrane inserts and by seeding different cell types onto it. We can use then the cell culture medium so to serve us as the circulatory system in terms of to bring the nutrients to the tissues. And importantly, those transfer membrane inserts enable us to cultivate the model at the air-liquid interface, which is for the lung compartment very important. So preparation of the model starts with isolation of primary blood-derived monocytes. So we obtain those mononuclear cells by purifying the blood samples that we obtain from regional transfusion centers. So we purify those samples using a cell density gradient. After this, we obtain, you see my cursor or not? Yes? Oh, all right. Just I'm trying to get the
cursor. If you want to highlight something, you go to the button at the third button from the left side. You see your cursor, and you can select, for example, the pencil to draw things.
The pencil. All right, I have it. Thank you. So here we can see this buffy coat. This is a layer of white blood cells, which comes after the radiation separation, which comes just below the plasma and above the the platelets and the red blood cells. Then we use magnetic separation cell sorting, anti-CD14 magnetic beads, so to obtain from the sample monocytes. These are the CD14 positive cells. And straight after the isolation, we start with differentiation of the two immune cell types that we're using in the model. And these are macrophages and dendritic cells. We cultivate them in specific growth factors in the medium for 6 days. And already during this differentiation period, we start seeding our epithelial cell line. So also here we selected A549s for this model, and we seed them on transfer membrane inserts, and we wait until they form a monolayer like this. On the day 6, we then assemble the co-culture by adding differentiated macrophages on top and dendritic cells at the bottom of the system. Importantly, we cultivate this, as I said previously, at the air-liquid interface with the aim that those epithelial type 2 cells, which A549s are, that they produce surfactants. This is very important, especially when investigating effects of inhaled materials like nanomaterials onto the tissue barrier. But I want to point out that also other cell lines can be here included, or cell types, also like HLV. Yes, we tried, and also bronchial cell types. But then we just need to make sure that we consider all the cell type specific properties then. I would also like to stress out that for this isolation and differentiation of primary human monocytes, we have now optimized the protocol so that we freeze directly the monocytes. We freeze them, and then when we need them, we thaw them and we differentiate them into the two immune cells. And this very much facilitates the experimental setup, so to not to always depend on the regional transfusion center. So this is now the model from a closer look. We can see A549s in the middle, macrophages on top, and dendritic cells on the bottom. I want to point out here that this is the model that is this is a very powerful tool to investigate effects of different compounds from either deriving from the apical or even from the bottom part to the model. But this model does not represent the airway blood area, because for this we would need endothelial cells, meaning the cells that present the blood vessel cells. So with this model, we do not specifically study transport of the compounds through the airway blood barrier. However, we still consider that cell culture medium is our source of nutrients. So yes, it brings the nutrients to the model. And also the model then secretes, all the cells secrete cytokines and other markers in cell culture medium, and then we can evaluate this from this medium. Here is just an insight into the immune cells that we use. I wanted to show you how macrophages and dendritic cells look under phase contrast microscope after 6 days of the differentiation. And we can also see that when we render confocal images, that pre-stained macrophages and dendritic cells are very nicely integrated on the model on top of the gray epithelial cells. All right, so all the experimental details are presented in our recently published paper in JAVE, and all along with the video where experimental details and especially those tricks and tips are presented in the video. So if you're interested, I would very much welcome you to have a look at the video, and then you can also drop me an email if you still have any specific questions. So now we have the model, and we will come back to it shortly. But before that, I would like to touch the theory about inflammation. So as I said at the beginning, I will present a diseased model, inflamed model. So why is alveolar inflammation so important? Either chronic or even acute inflammation, it occurs in a variety of lung diseases, such as in fibrosis or in chronic obstructive pulmonary disease. And it's important because when alveolar tissue is inflamed, this really impairs the gas exchange in the lungs. And this then also affects that less of the oxygen or not enough oxygen might be derived to other organs. Inflammation occurs as a consequence of a stimulus or a combination of stimuli in the lungs or in the whole tissue, in the tissue. And an example of such stimulus is LPS, lipopolysaccharide. This is a component of gram-negative bacterial cell wall. And also in vitro, we can use this compound to mimic inflammation. But I want to point out that just the presence of stimulus in the tissue, it does not always mean that the tissue will be inflamed. There is first a series of reactions that occurs, and this is called the pro-inflammation, which is the state of alert in the tissue, and the most significant characteristic of of the pro-inflammatory cascade of events is relation of pro-inflammatory markers. For example, such as chemokine interleukin-8 and tumor necrosis factor alpha as the main inflammatory factor, or IL-1 beta and many others. And in vivo, this also means that such a state attracts then immune cells from circulation so that they come at the site of infection, so in alveoli. So as I said, in vitro, we can also use this tool. And if this stimulus LPS, and if we're lucky, then our model will respond to it by showing pro-inflammation specific characteristics. So how to include this in our experimental setup? After the model had been at air-liquid interface for 24 hours, we can challenge it with LPS. And we optimize this in our work so that LPS can be applied either from the apical side or from the basal side of the model. So if we start with the apical side, so this means from the top of the model, we apply LPS only as a thin layer of dispersion on top of the model. Why is this important? So that this liquid really evaporates so that air-liquid interface conditions are preserved. Again, I'm pointing out in lung research, this is very important. This type of LPS or inducing inflammation with LPS comes in very handy when we want to test some therapeutics that we would deliver in a medium, so to resemble a therapeutic being delivered from circulation systemically, because we would like to avoid a contact or possible interference between a stimulus and the investigated compound. On the other side, when we apply LPS from the basal side in the medium, this comes in handy when we're testing, for example, respiratory hazard of materials, which we're interested in today. So for this is good because then the materials we would apply directly from the top, for example, as Kirsty presented, using an aerosolization system. All those details and about the meaning and the methodological steps how to induce both the types of inflammation are presented in our recently published paper, and please just contact me if any additional information would be required. So regardless of the approach, an inflamed tissue has its specific features. So one thing that a pro-inflammatory status in the tissue does is that barrier integrity is compromised to a lower or higher degree. A compromised barrier integrity can be visualized using confocal microscopy, for example, or also using permeability tests such as fluorescein or even TR, as we discussed before, but not for apoptosis. And as I mentioned before, a very general characteristic is secretion of pro-inflammatory markers. And we can assess this with ELISA, and we can also do the respective gene analysis with PCR. Also, various other endpoints can be or are even advised to be more monitored in parallel, such as cytotoxicity, oxidative stress, and many others. But with this, I will not go into details. So as a first thing, I wanted to show you that basal site of application, meaning applying LPS in the medium, creates a more prominent pro-inflammatory conditions in the tissue. It makes sense because this really resembles a systemic inflammation. We can see here with those white circles in the middle panel that applying LPS for 24 hours in the basal compartment, it creates those holes in the tissue, which we can see here where cells are marked, actin is marked, cytoskeleton in pink and nuclei in blue. And we can see that these are here some empty spaces, which are less prominent when LPS is added to the apical side. And here nicely, we can also see at some places macrophages that are colored in white, and here is one dendritic cells that is green. It's a bit faint, but it is there. And then also, as I mentioned, we can use cell permeability assays. So here we use the labeled dextran. And we can see where LPS is applied in the in the basal site. This is marked with 1. We can see that the effect of cell layer permeability was comparable to the one where we use EDTA as a positive control. Whereas when we applied LPS apically, this was much more similar than to untreated cells. As I mentioned, also cell viability is good to be tested, and we can see that both the approaches actually did not cause any impaired or did not induce cytotoxicity, which is good because we want to measure the events before that. So we don't want LPS to really be toxic at the end of the day. And then we also tested secretion of pro-inflammatory markers. Here I'm showing an example of IL-8 and TNF-alpha. And we can see that with both the approaches, there are elevated levels of those proteins secreted and also of the genes, of the respective genes. We cannot really say, and it was not statistically significant, that the approach 1, this means when applying LPS in the basal compartment, would trigger a higher response, which is okay, good, because it means that also when LPS is applied apically, it actually works. Both the approaches work very well. So now we have an inflamed model, and now it's ready to exposures to nanomaterials. So this is something that we have not yet done in the lab, but it is on our to-do list in the group. So this means that after we have the LPS challenge, the LPS is applied in the system, for example, in the basal compartment for 24 hours. We would then here do the tested material. Here would be the exposures on the day 9, and then, for example, on the day 10, which would be 24 hours of exposures to a nanomaterial in an inflamed model, we would then do sample collection and data analysis. I would personally recommend that LPS here throughout the period would stay in the system because this would more realistically mimic the conditions that we have in the lungs. So once we have a systemic inflammation, if we don't get the treatment, most likely it will not faint very fast or go away very fast. So here I'm coming already to the end of my presentation. So what I presented today is that we can, in 10 days, we can assemble the model from primary immune cells and a cell line, and we can induce inflammation and expose to nanomaterials. And of course, we need to analyze the whole thing. And I want to point out that LPS is a very good pro-inflammatory stimulus for such a model in vitro. And the main characteristic of pro-inflammation can be then tested with very widely accessible tools such as confocal microscopy and permeability tests for impaired barrier integrity, and then ELISA and PCR and other bioanalytical techniques for the other markers. With this, I would like to thank my supervisors at the University of Fribourg and the whole Bionanomaterials group. And I would, of course, like to thank the Petrels Project's partners and the funding for all the support. Thank you for your attention, and I'm happy to take any questions. Yes, the link to the video, I can post it after I finish all the questions. I will post it here in the chat. Is that all right? Yes, okay. We had a look at CD86 only, but not 54, thank you. But we had a look at CD86 only in monocultures. We haven't optimized flow cytometry approach yet to the whole triple co-cultures because it gets quite complicated. But thank you. Okay, interesting question about the surfactant. It might be, but it's also the fact that it's much more LPS, like the nominal dose is higher. But might be that that surfactants do protect from LPS from the top. It might be, however, I think LPS is quite soluble, so it should also go through the surfactant and come to the cells. I think so. For confocal microscopy, we fix it with PFA, and then we stain with immunofluorescence dyes. I can send you the link to the paper where we describe it, but it's quite a simple approach, or the widely used approach. Yes, I see now. Yes, all right. I do agree, might be. Good point, thank you.
Well, we saw quite a couple of questions. Thank you very much, Barbara.
Thank you.
Happy now to add on to Rui-Wen. And I will quickly upload the presentation. With regards to optimization of an air-liquid interface co-culture model to estimate the hazard of repeated aerosol exposures. So please, Rui-Wen,
hello.
Continue with the presentations. Thank you very much.
Okay. Hello everyone. My name is Rui-Wen from IVM in the Netherlands. Today I'm glad to introduce the optimization of an early cell co-culture model for aerosol exposure. As we know, human bronchial epithelial and alveolar epithelial cell models are both options for in vitro experiments, especially to ultrafine particles, since both of these two regions can receive ultrafine particles on their surface. As the two presenters introduced the alveolar cell models before, today I'm glad to introduce one co-culture model. Oh, sorry. I'm glad to introduce the optimization of an epithelial plus macrophage co-culture model under early conditions. And then I will show the application of this co-culture model for early exposure to nanoparticles. Here are some backgrounds. As we know, the inhaled particles can deposit onto the lung bronchial epithelium. So our study focused on the lung bronchial epithelial cell models to evaluate the inhalation exposure. To closely mimic the inhalation exposure, the early culture and early exposure were developed. So during the early exposure, the cells can be directly exposed to the various substance. It's real-time exposure, and there is no loss during the exposure, and it's also what happens in our lungs. But if we want to do the early exposure, the cell selection is necessary because not many cell types can remain viable for long-term early culture. In our studies, we selected Cal3 cells, 16HB, A292, and BIS2B cell lines for testing. As the epithelial cell for the epithelial cell models, the barrier function and single-layer morphology are essential criteria. So we measured the TR values of these cells and also did the Z01 protein stainings. For all of these cells, the cells were cultured under submerged for 7 days. Afterwards, we removed the apical side medium, and then the cells were started to culture under the early conditions. So from these results, we can see only the Cal3 cells can maintain the monolayer structure and keep a strong tight junction under long-term early culture. So from the results, we can see only the Cal3 cells can maintain the monolayer structure and keep a strong tight junction under long-term early culture. Also, the macrophages play an important role in uptake and clearance of particles in our lungs. So, to evaluate the realistic responses to particles, the co-culture models need to be created by adding macrophages on top of the epithelial cell layer. In our study, to build the Cal3 and Cal3 plus MDM co-culture model, we first differentiated the macrophages. We first differentiated the primary monocytes to macrophages by adding the MCSF. Afterwards, the differentiated macrophages are added onto the Cal3 cells layer for several hours for adhesion. After removing the apical side of the medium, the co-culture models are ready for testing. But during our experiment, we found the macrophage adherence time and seeding concentration can affect the monolayer structure and sensitivity of co-culture models. In our experiment, we tried the different time points and seeding density for macrophages adhesion. We found a long-term period, for example, 24 hours, with medium on top of the monolayers, can affect its structure. So to avoid this negative effect onto the monolayer, a short adhesion period, for example, the 4 hours, should be used. However, the efficiency of macrophages adhesion decreased with a short adhesion period, then decreasing the sensitivity of co-culture models. To make it balanced with our optimization, we decided to use the seeding density at 50,000 macrophages per square centimeter at the 4 hours adhesion. We also did the mutual comparison between Cal3 monoculture, Cal3 MDM, and Cal3 TDM models after exposed to LPS. So from the results, we can see the Cal3 MDM model shows the higher sensitivity in inflammatory responses to LPS exposure. Next is the application of Cal3 MDM model. We use this model for a single day exposure to nanoparticles, DQ12, with the early exposure system. So here are the information of the parameters. So after exposure, we found the Cal3 plus MDM co-culture showed the cytotoxicity and the inflammatory responses to DQ12 exposure. But in our parallel experiment, the cytotoxicity and the inflammatory responses was not induced in Cal3 monoculture by DQ12, and the repeated exposure experiments to DQ12 and other nanoparticles are still ongoing. So based on our results, we can draw some conclusions. Cal3 MDM co-culture model had the epithelial monolayer integrity on the long-term early culture, and it showed the increased sensitivity in response to LPS and DQ12 exposure. Therefore, Cal3 MDM co-culture model is a good option for early exposure to aerosols for toxicity testing. That's all. Thank you.
Thank you, Rui-Wen.
Yes, thank you. Sorry for the
mistake. You could come back with your presentation. Do we have some questions? At the moment, I cannot see questions in the chat box.
Okay.
So I thank you again for the moment, and I will give the word to.
Yeah. Okay.
There was one. Just a second. Put it on display.
Yes, yes, we use the primary monocytes, and also we use the THP1s macrophages as a comparison.
Okay, great. So might be one more. Okay, now if there are no urgent questions at the moment, I would.
Sorry, can I add some information?
Yes, please, sure.
As this protocol, the cultural protocols, including the co-culture protocols, were also published recently. So if you have any questions, or if you are interested in it, so please let me know. I can send also the links to you.
Thank you very much.
Thank you.
Very informative and very important to also promote these models, that more people get access to and start working with them to become more realistic in their models. And I'm very glad now that we have another presentation of a model which is in a different organ, the intestine model, why it is a challenge for in vitro research. So Angela, you're gonna let us know. Thank you for being here. So you still need to unmute yourself, your microphone. For unmuting, you find the button. It's a red button underneath the Nanosafe 20 sign.
All right, is that in the wrong part of the screen? But is the presentation still there? It's
yeah, now it's gone. Somebody removed it. Okay, please.
Okay, brilliant. Thank you very much for the opportunity to present something about an organ that is, well, usually a bit forgotten, let's say. So I'm very happy to be in this prominent liver-lung sandwich of patrols. The first couple of slides, I will focus on setting the scene a bit for the organ, for the intestine itself. Hopefully, it also becomes clear why it is such a challenge to mimic the organ for in vitro research. And then in the second part of the presentation, I will focus on the in vitro model that we are working with at the IUF, where I'm currently working as a postdoc. So just a couple of basic information, as I'm pretty sure that most of you have heard quite a bit about the intestine already. It is the largest part of the orogastrointestinal tract, and it's located mainly in the abdominal cavity of the human body. It is roughly divided into small and large intestine, and probably best known for its functions in food digestion and nutrient absorption, which is mainly taking place in the small intestine, as well as the regulation of the water household, which is mainly done in the large intestine. However, apart from this, it is also a very important contact and entrance organ for nanomaterials, as different types of nanomaterials can be, for instance, incorporated in food products or beverages. They can be part of pharmaceuticals, but also inhaled particles can be cleared from the airways and then swallowed afterwards, thereby getting in contact with the gastrointestinal tract again. However, I would really like to point out here that it's just a lot more than a weird tube that transports your food and magically absorbs nutrients, but it really is an organ of superlatives. It is forming one of the largest interfaces between the organism and the environment, the outside environment, which can be seen here in this cross-section that I've included as an example. So the three-dimensional structure of these folds and the microvilli that are formed on the enterocytes is resulting in a vast surface area. Therefore, it is also not surprising that it's one of the largest immune compartments of the human body, because this very critical balance of allowing nutrient absorption across the epithelial barrier while keeping out potentially harmful antigens needs to be guarded somehow. It also harbors the largest population of bacteria, the gut microbiota, and what is probably not so well known is that it contains a vast nervous system, the enteric nervous system. The gut is often called the second brain, and this is not so surprising anymore once you realize that it actually is the largest collection of neurons and glia outside of the brain. A couple of words to the structure and the anatomy of the small intestine. So, as said before, it is kind of just a long tube of several meters in length, and it is lined with a monolayer of epithelial cells, which fold into crypts and villi, increasing the surface area of the whole organ. The main cell types forming the epithelial barrier are enterocytes and goblet cells, which make up more than 90% of the cell types. The goblet cells are mainly known for the mucus production, which forms an additional, very important physical barrier lining the whole epithelium. The intestinal mucus is also important for the gut homeostasis in itself and also provides an important living environment for the microbiota. Apart from the epithelial cells, we have representative cells or representation of the immune system. And here we have to distinguish between aggregated immune cells or immune compartments and isolated immune compartments. The Peyer's patches, to which also the famous microfold or M cells belong, can be counted to the aggregated immune compartments. And individual cells, as for instance, intestinal macrophages or also dendritic cells, are isolated immunocompetent cells. However, apart from this vast diversity of cell types, there is a lot more that needs to be considered when looking at the gastrointestinal tract, because you have an ever-changing environment depending on the kind of food you're taking in or whether you are actually in a fasted or in a fed state. There is a huge variety in the different digestive fluids, starting with the saliva, going to the gastric acid, in the stomach, and then the intestinal juices starting from the duodenum. For instance, encouraging a heavy change in the pHs depending on where you actually are in the gastrointestinal tract. You have the previously mentioned mucus and other digestive components like, for instance, enzymes. And then later on, going to the large intestine, a huge number of commensal bacteria, which can also influence the way that the intestine is reacting to, for instance, chemicals, pharmaceuticals, but also particles. So we have this multitude of individual factors which can be considered, but do not necessarily have to be considered in an intestinal in vitro model. The problem is that we actually still don't know yet which of these factors are essential to build a representative and a realistic model, and which are just nice to have, or which may be important or may be essential for addressing very particular questions. The problem is that each of these factors, of course, increases the complexity of a model, and this can be both positive and negative. It can be positive in the sense that you increase complexity and thereby make your model more closely associated to an in vivo situation. But on the other hand, the complexity of a model is always inevitably related to the robustness of a model and its reproducibility. So the more complex you make a model, the more difficult it will be to set it up and obtain the same results in a different lab. If you have a look at the literature, there is really highly sophisticated in vitro models of the intestine out there, and the majority of them have been developed in the last 10 years. This includes, for instance, an involvement of a three-dimensional structure, the inclusion of flow, even the inclusion of microbiota, which I think until a couple of years ago, nobody would have expected to be possible. But you also see that there is actually very, very little application of these highly sophisticated models in applied research. So we still have to see where the development of intestinal in vitro models will take us in the future. So after having set the scene now, I would like to focus on a model that we are working with at the IUF, which has been developed, or actually advanced in patrols. I call this the macrophage model, as opposite to the M-cell model, which was used by other partners within the project. To remind you a bit of what the major pillars of intestinal research are at the moment, we have kind of the gold standard, the use of human materials, for instance, generated from biopsies. But unfortunately, within Patrols and at the IOF, we do not have the opportunities, the possibilities to really get to this kind of material. So we are focusing on the other two big pillars, the use of animal models on the one hand, but then also the further development and advancement of in vitro models. So as just mentioned, in Patrols, we are focused on two different types of models. On the one hand, that is the so-called M-cell model. It was a triple culture setup of CACO2 cells, the E12 subtype of the HT29 MTX cell line, as well as Raji cells to induce an M-cell formation of the CACO cells. This has been done, Heriot Watt, as one of the partners in Patrols, has focused on this type of model, but unfortunately nobody of them could actually make it today. But if you are interested in this, if you are interested in receiving more information on that, then please let me know and I can give you a contact to Heriot Watt. At the IOF, we have focused on the triple culture, also on the triple culture model of CACO2 cells and HT29 MTX cells, but we have focused on the introduction of an immunocompetent cell line, and here we have used THP1 cells as a model for macrophages. The important part about this model is that it can be used in both a healthy state, which I'm calling stable, or in an inflamed-like state. The macrophage model has been advanced in Patrols from a previously described co-culture model of CACO2 and THP1 cells. This model has been described in detail in a publication a couple of years ago. And if you would like to know more about the seeding densities and why the parameters have been chosen the way that they are still actually used today in this triple culture model, then please check out the manuscript, because you will find a lot more information in there than I can give you now in this presentation. The predecessor model was now in Patrols advanced by including the HT29 MTX cell line as a model cell line for goblet cells into the epithelial transwell layer. So therefore, we are here talking about a triple culture model of enterocytes, goblet cells, and a macrophage-like cell type. And when setting up this model can actually be divided in three individual stages. The first stage is focusing on the epithelial co-culture that is seeded onto transwell filters. Here, CACO2 and E12 cells are seeded in a 9:1 ratio and then maintained for 21 days in culture to allow for barrier formation as well as mucus secretion. What is happening over these 21 days, you can actually see in the graph to the right of the scheme. The barrier integrity is continuously increasing, which leads to an increase in barrier resistance, here measured as transepithelial electrical resistance. What we also see is that a very dense cell layer is formed where you can actually still identify the E12 cells within the majority of the enterocytes, within the majority of the CACO2 cells, which is these larger islands formed in the image to the right of the graph. The second part that we are waiting for is the release, the secretion of mucus, which we can detect either by specifically staining for particular mucins, as shown in the image at the bottom on the left for mucin 5AC, which is the major mucin secreted by the E12 cells, or more generally staining neutral and acidic mucus, as here shown by the PAS and Alchem blue staining. The second stage of the model development focuses on the THP1 cells. This is done 24 hours before the start of the co-culture. THP1 cells are treated with PMA for 24 hours. We are doing this in small culture flasks before they are then detached using Accutase and reseeded in transwell-suitable 12-well plates. Why are we having to treat the cells with PMA? So one of the most obvious changes that is induced can be seen in the images to the right of the scheme. We have a change from a suspension cell line to an adherent to an adherent cell type, which also is characterized by a very significant change in morphology. But which is a lot more important for us is what can be seen in the graphs below the scheme. It is the change in pro-inflammatory cytokine release that is induced by the PMA treatment. In the left graph, we see the cytokine release of interleukin-1 beta, interleukin-8, and TNF-alpha in response to lipopolysaccharides without PMA treatment, compared to the right after PMA treatment. And you can see here that especially the release of interleukin-1 beta and interleukin-8 is increased about 10-fold with the PMA treatment. And this is the amount, or we really need this high amount of pro-inflammatory cytokine release to be able to induce an inflammation-like state in the co-culture. The last stage of the model development is actually bringing these two first stages together. So the fully differentiated epithelial transwell cultures and the PMA-treated THP1 cells. Once you have the reseeded THP1 cells, you still have to decide whether you want to actually start the stable culture or whether you want to use the inflamed model. If you focus on just the stable or the healthy model, you can directly proceed by placing your epithelial transwell cultures onto your THP1 cells and you're done. If you want to use the inflamed model, the THP1 cells first have to be activated with a cocktail of lipopolysaccharides and interferon gamma, which has to take place for about 4 hours before the epithelial transwell cultures can be placed on top. Once you have the triple culture set up, you can maintain it for an extended amount of time. We have tried everything from just 24 hours up to 6 days without exposures and 5 days with exposures. This transwell setup, and especially the 12-well format, offers you the great opportunity to have a vast amount of different samples that you can probe. For instance, the supernatants from the apical and basolateral compartment can be collected separately or can be collected individually. And the transwell filter is actually large enough so that it allows you to cut it in half, use 1/2 of the cells for a direct analysis, for instance, protein lysis or protein arrays, but also RNA analysis or DNA damage analysis with the COMET assay. And the other filter half can be fixed and used for stainings later on. There we go. Brilliant. I know this is a lot of graphs and I only want to go through them briefly because the first thing that we had a look at is, of course, the characterization of these two states and whether we are able to distinguish them without any doubt. So what I can say is that, yes, both of these models, or both of the states of this model, show very distinct features. Starting from this first graph here on the right of the text, we have the barrier integrity, which is temporarily reduced, temporarily impaired for the inflamed model, whereas for the stable model, it's all relatively, well, stable actually. Going further to the right, we have the LDH activity as an indicator for cytotoxicity. And we see that in the inflamed model, we have a continuous increase of LDH activity in the apical compartment, showing or suggesting that we have an ongoing cytotoxic effect of the strong inflammatory stimulus that is going on. At the bottom row, we have a very particular mucin profile, most notably a significant reduction in mucin 2 and mucin 5AC, as well as a very substantial increase in mucin 1. What is interesting about this mucin profile is that it's relatively comparable to the mucin profiles in patients with inflammatory bowel diseases, where you also see an incredible increase in mucin 1. These two graphs belong together, showing the cytokine release in the basolateral compartment of the stable triple cultures as well as the inflamed triple cultures. We have on the left side, the stable cultures, and we see that interleukin-8 is present, but at relatively low amounts. And for the inflamed model, there is a huge release of pro-inflammatory cytokines. These are only examples. We have looked at a lot more, and there's a vast area released by mainly the THP1 cells. Coming now briefly to application possibilities and later on also some limitations. What can the model be used for? We have so far used it for acute as well as repeated exposure studies, mainly using different types of nanomaterials. The model is stable for at least 144 hours, and even with repeated exposure studies, where we exposed the cultures for 5 days in a row, we managed to keep it working for at least 103 hours. We have also used the model for soluble compounds, for instance, diclofenac as a chemical control, but yeah, as said before, mainly sedimenting particles. The model can be used to study the effects in a healthy tissue, but also to have a look at whether ongoing inflammation is actually having an impact on tissue susceptibility. We have shown this with a predecessor model already and published it earlier this year, that ongoing inflammation actually enhances the toxicity of silver nanomaterials. And we hope, of course, to see a lot more of this happening in the future. Unfortunately, there is also some limitations, but I guess that holds true for most of the models. It is not suitable for low-density particles. That is actually self-explanatory because we are exposing in a submerged culture. So therefore, we rely on either substances that are soluble and therefore get in contact with the particles, with the cells, or high-density particles that actually sediment over a reasonable amount of time. If the particles are too low density and do not sediment, they will not get in contact with the cell layer, and therefore no effect is to be expected. Also, due to the lack of M cells, the particle transport is likely underestimated. So I would not recommend this model to actually, if you want to focus on particle uptake and particle translocation. It is also probably not capable, the model that is, to detect pro-inflammatory effects, which may sound a bit strange, keeping in mind that we have an immunocompetent cell type in it. But the problem is shown here in this graph at the bottom left. There is a very strong controlling effect of the epithelial cells over the THP1 pro-inflammatory response. What this graph shows is the release of interleukin-1 beta, interleukin-8, and TNF-alpha in response to LPS if THP1 cells were exposed in monocultures or, however, exposed in co-cultures. And what you see is that the release of all three cytokines is significantly reduced, but especially interleukin-1 beta and TNF-alpha are almost absent. And the problem is that these two cytokines are absolutely crucial to get the inflammatory response going. This controlling effect of the epithelial cells can partially be taken care of, can partially be reduced, but you need a really, really strong stressor. Here, for instance, we used EDTA to entirely disrupt the epithelial cell layer. And then there is an opportunity for apically added lipopolysaccharides and interferon gamma to sediment or to translocate to the basolateral compartment and activate the THP1 cells later on. However, this is also not always working because we have tried the same with diclofenac, which also destroys the cell barrier entirely, but there LPS and interferon gamma did not have any effect. So with this, I would like to thank the organizers for giving me the opportunity to present this to you, and also, of course, to Patrols for providing me with the funding to continue working on the in vitro models, and especially with a focus on the intestine, which hopefully will receive a bit more attention in the future.
Thank you very much, Angela, for your great presentation. I do try to find questions for you in the chat box, but so far I have not seen any. But it seems obviously that the more realistic such models become, a little bit less susceptible towards pro-inflammatory stimuli, as you were.
Yeah, absolutely. They become.
It could be. I mean, if one carries it a bit further to even including microbiota, and so could then become even more, or even less susceptible, so more robust. What would be your guess?
I think so too. So what we have seen recently, and it is an accepted publication, which hopefully soon becomes available in small, we have compared different in vitro models of the intestine, having, for instance, proliferating monocultures, then the epithelial co-cultures in themselves, and then the triple cultures together with the THP1 cells. And we have actually only seen effects of different types of particles, nanoparticles, in the proliferating monocultures. But already once you have an epithelial co-culture that has differentiated cells, that is a complete cell layer, let's say, a dense cell layer, then the reaction to at least very obvious or easy to detect endpoints is almost zero. So I think you would really have to go on very sensitive endpoints, like going onto the proteomics level or transcriptomics level to still see something there.
And the other point, which is also hitting the GI tract, is probably also the chronicity of such kind of things going on, not just up to 6 days, but even for longer times, isn't it?
Exactly. So in the beginning of patrols, we have discussed, or there was the idea to also expose the intestinal models for up to 14 days. We have decided against that because the turnover rate, the cellular turnover rate in the intestine is so high. So you actually have an average, a maximum lifespan of your typical enterocyte for 5 days. And therefore we have left it at 5 days for now. But of course, it might be important in the future to still extend that.
Okay, thanks a lot. There is also a question from the audience. I put it on display.
No, we have not, because primary small intestinal cells, at least to my knowledge, I'm not sure at the moment whether there is anything commercially available which has been treated one way or another, but using primary cells isolated from intestinal tissue is not as straightforward and not as easy to do as with other tissues. So you always rely either on the use of the mesenchyme or the introduction of it, or you really need to focus directly on the use of intestinal stem cells and develop intestinal stem cell organoids from that. And that is just too much effort for us. These protocols are too complex to do it on the side. You would have to fully focus on that and leave aside any other type of in vitro model for that. Yes, they are differentiated for 21 days.
Okay. Thanks for the questions. So definitely things can go on in the chat.
Yeah.
There was a little bit of delay, of course, with this online meeting, so maybe there came another one.
Do I still have time to answer that one?
Yeah, sure.
Okay, perfect.
I don't want to accelerate. I was just seeing no more questions.
Okay. Yes, we have. We are. This is one of our one of the research focuses actually that we are following. So we are, as a standard, always including an artificial digestion of the nanoparticles. For the last 1.5 to 2 years within patrols, we have actually always run a side-by-side analysis. So we compared whether this artificial digestion is changing the effect of that nanoparticles have in these intestinal models. And we are now actually focusing only on the on including nanomaterials after an artificial digestion.
Also very relevant point. Mm-hmm. Yes. Of course, we can always keep discussing in the chat. This is an advantage of online meetings. If there are no further questions now, we probably put it to some chat discussion between Angela and the other attendees. And I would then like to move to the next presenter, Samantha Llewellyn. And I will put on her presentation. Actually, she provided a film again. And then she will, of course, be available for any further question.
So welcome, my name is Samantha Llewellyn, and I'm going to be talking to you today about advanced engineered nanomaterial exposure regimes for more realistic dosing scenarios on 3D liver models in vitro. So as a bit of an overview, the liver is the main detoxification centre of the human body and is one of the only organs that possess the capability to regenerate. In addition to this, it is the main site of secondary nanomaterial deposition following either primary routes of exposure, so ingestion, inhalation, injection, and dermal penetration. The liver itself contains of two main lobes, both of which have 8 segments that embody thousands of liver lobules. These lobules, as shown in B, are hexagonal structures constructed by repeating segments radiating out from the central vein. So each segment, as shown here, links to one another by the liver sinusoids and consists of the portal triad, so the bile duct, portal vein, and hepatic arteriole. Within the liver, there are two main cell populations, parenchymal and non-parenchymal cells. Approximately 80% of the liver volume is occupied by the parenchymal hepatocytes, whilst the remaining non-parenchymal fraction is made up of endothelial cells, stellate cells, and the resident liver macrophages, Kämpfer cells. These cells work together to maintain metabolic homeostasis and detoxify any xenobiotics. So based on this and the principle that 2D cultures do not encapsulate the intricate multicellular interactions and metabolic activity observed in vivo, as well as their 3D counterparts, we began to develop a 3D cell line-based liver model. So the aim of this work was to advance existing 3D in vitro liver models to enhance their physiological relevance and develop a liver model able to retain both its proliferative capabilities whilst remaining viable for 14 days in culture. So this was to allow for long-term and repeated nanomaterial exposure regimes to be evaluated, but also with more of a primary toxicological focus on genotoxicity endpoints. So our model itself, as depicted here, is established using the hanging drop method to form 3D liver spheroids, which consist of about 4,000 HepG2 cells per spheroid. This equates normally to about 500 micrometers squared in diameter, and they are available for over 10 days in culture. So in preparation to build this model, there are a few key components you'll need to bear in mind. This model is based on the HepG2 cell line from ATCC, and if you're looking to do a genotoxicity-based study within this model, I advise that you run a preliminary experiment on the cell line first to assess the background genotox prior to then diving into your research, as some supplier stocks do have a higher background than others. So in addition to this, and believe it or not, the 96-well plate you use is important, as the lids on some of the plates can be looser than others, which makes it more difficult to form full plates of spherical shaped spheroids. And then most of the reagents will be prepared on the day of, but the 1.5% sterile agarose gel will need to be prepared in advance. On the right, we've got a little bit of a kind of overview of the breakdown that will to be expected and what will need to be done on particular days. And this entire process can take anywhere between 10 days to 2 weeks, depending on your nanomaterial exposure length. So once we developed this model, we did some baseline characterization work to see how the model performs and compares to other models on the market. So we found this model remained viable for over 10 days in culture and was shown to maintain, if not increase, albumin and urea production throughout the 14 days in culture. This highlights that the model remains functional throughout the period. So as most spheroid models, the development of the necrotic core often limits the longevity of these proliferating models, as the larger and more compact the spheroid becomes, the harder it is for the oxygen and nutrients to diffuse through. Some models, like the primary human hepatocyte models, offer a longer culture period as they are smaller, but limit the evaluation of fixed DNA damage as they are non-dividing static models. So it does completely depend on the specific toxicity endpoints you wish to study. However, I shall take this opportunity to take you through the development of the 3D liver model that we have developed and its particular applications. The methodology I'll be explaining or showing to you has also been published in Jove, and there is a video coming soon to support that. And in addition to this, we do have a co-culture model available, but this characterization data is yet to be published. Okay, so the process begins with seeding the 3D liver spheroids, and in short, we seed 20 microliter drops of Hep2 cell suspension onto the lids of the 96-well plates. I have prepared a little video to help visualize this process. I'm not sure how well this will come across online, but we'll give it a go and I'll talk you through it a little bit. So initially, we add PBS to the base of the plates to ensure, or to maintain hydration and ensure that the spheroids do not dry out during the 3-day formation process. You need 100 microliters of PBS, which will be added to the base of the plate. So once this is completed, you'll take a cell suspension of about 2 × 10⁵ cells per mil. This will equate to about 4,000 cells per spheroid in each 20 microliter drop that gets added to the surface of the lid. So when adding these drops, take care to make sure the spheroid droplets are actually added in the center of the grooves, as you can see there. As this will make sure that the spheroids form directly in the center of the drops. For each plate, about 5 ml of cell suspension is required in order to seed the full 96 wells. Once you have seeded these, you then flip the lid and place back onto the bottom of the 96 well plate. This can be quite tricky and does require a lot of practice, because sometimes whether you go too quickly or too slowly, it can affect how the spheroids then stay on the lid of the plate. Okay, so what to expect? In most cases, not all 96 spheroids seeded will make it to the day of exposure. Some are lost during either the lid flipping stages, or others during transport to and from the incubator, or even whilst forming over the 3 days in the incubator. A common cause of the misshaped spheroids can be just from the disturbance of going in and out of the incubators aggressively. So it might be worth putting a note on the outside of the incubator just to inform other colleagues that may also be using that incubator. So as shown here between D and F, and then highlighted with a green outline, is a good quality spheroid. So it's what you'd expect to see. So you want a compact, singular spherical structure that hopefully is formed over the 3 days in the hanging drop. Or you could have poorly formed spheroids as shown in G to I and highlighted in the pink, where you've got protrusions and it's kind of maybe a little bit stuck to the bottom of the well, or you've got bits of cells that have broken away from it, or even individual kind of independent spheroids that have also grown within the well itself. So ideally, if this happens, we don't tend to use those spheroids. So yes, you kind of want them to look very similar to kind of D and F really before you proceed. Following the 3 days in culture, we transfer the hanging drops to the base of the 96 well plate, pre-coated with the 1.5% agarose gel, as shown in the following video. So to do this, you carefully remove the lids and you then empty the PBS out of the base of the plates. I tend to use just a bucket to empty all the PBS out, and then tap any of the remaining liquid out using just some glue roll. You then microwave the agarose gel to make sure it's molten, and use about 50 microliters of the gel to the bottom of the 96 well plate. I normally leave it for a couple of minutes before adding any media to the top of this, as you need the gel to set first. So once set, you can add 100 microliters of media to the plate, and then we just replace the lid back on top. You then take the 96 well plates and put them in a plate centrifuge, spinning them for 3 minutes at 1000 RPM. And these should then cause the Hep2 spheroids to drop from the lid of the plate to the base of the wells, as shown here. You can see the little individual spherical dots in the center of the wells. So after this, we allow the spheroids to settle for 24 hours before we expose them to varying concentrations of engineered nanomaterials. Prior to the exposure, these materials are pre-wetted in 95% ethanol and sonicated continuously for 16 minutes in 0.05% BSA at 21 amps, as described in the NanoReg dispersion protocol. All the materials we have used have been provided by the JRC with the exception of silver, which is from Sigma. Our aim was to assess the toxicological effects of low-dose, longer-term E&O exposures, and so the concentration range that we generally work with is 0.2 micrograms per mil to 10 micrograms per mil. It is important to note, however, that these doses are prepared as a double concentrate to account for the 50% dilution when adding 50 microliters of the dose medium to the 96-well plate, which already has 50 microliters of culture medium in there. So I always make up my doses double concentrated. So following the exposure period, the supernatant and spheroids can be harvested simultaneously or independently if necessary. So for supernatant harvest, about 50 microliters of the supernatant is taken from the surface of the wells in order not to disturb the cells, whilst the full 100 microliters should be taken, spheroid included, if harvesting both simultaneously. So this 3D model is compatible with a number of biochemical endpoints as listed here, so from cytotoxicity and liver functionality to inflammatory markers and genotoxicity. Each plate can yield about 5 mils of supernatant, and so any of the assays based on supernatant, such as your liver function or inflammatory based ELISA markers, one 96-well plate will be more than sufficient. However, for cell-based assays, more plates may be required per dose depending on how many cells the assay requires. So one key feature of this model, though, is it is able to support fixed DNA damage assessment using the micronucleus assay, as it does remain proliferating throughout 14 days in culture. So this is just a few applications of this liver model. I would have gone into a bit more data and maybe shown you some of the results, but as this is more of a teaching module, I'll just show you some of the kind of applications that we've used this model for. A little bit of what does the liver see? So with the liver being a secondary site of exposure for engineered nanomaterials, following systemic translocation from the primary exposure routes mentioned earlier, it is highly unlikely that the liver will be exposed to pristine nanomaterials, as interaction with a variety of molecules in these different cellular compartments can transform the physicochemical properties, so surface chemistry, protein corona, morphology, dissolution of these materials, which can then affect their biological interaction and the toxicological output. So to mimic this potential biotransformation, we developed an SOP whereby the nanomaterials undergo a series of incubations in synthetic biological fluids that was aimed to then simulate the ingestion and digestion of nanomaterials specifically. So initially, the E&Ms are incubated at 37 degrees for 30 minutes in a gastric, so pH 2.7, simulant fluid, followed by 30 minutes in an intestinal fluid at pH 9.5. This is a protocol devised by Angela Kämpfer and her colleagues at the IUF in Germany. Following this, we then decided to add an additional incubation of 1 hour in human blood plasma prior to further dilution in media and exposure to the 3D liver spheroids. So this particular SOP or scenario mimics the gastrointestinal tract kind of ingestion exposure scenario to the liver. So furthermore, we had a look at the manner in which the E&Ms are dosed onto the cells within in vitro cultures, as this may influence their biological interaction, as humans are usually exposed to low doses of E&Ms over extended periods of time, as opposed to a single high dose as typically applied in cell culture practice. So with this in mind, we decided to address this by undertaking a fractionated repeated dosing regime, as well as a bolus single hit dosing regime for a longer term 120-hour nanomaterial exposure. So for the repeated dosing regime, the final concentration is fractionated into 5 equal concentrations and dosed daily, which will inevitably amount to the same single bolus dose applied on day 1 in the other regime. This is kind of depicted in the diagram here, where you can see the 5 micrograms per mil final concentration has been split into 5 1 microgram per mil doses over the duration of the 5 days. So on the day of harvest, we'll then look at the same toxicological endpoints, so the micronucleus assay, inflammatory markers using the ELISAs, and just general liver functionality as well, just to assess if this has made any difference in the way that you dose the nanomaterials. So to conclude, although with its limitations, this advanced 3D in vitro liver model is able to support a range of toxicological endpoints for nanomaterial screening. We've been able to develop an SOP for more physiologically relevant nanomaterial dosing regimes with a tiered kind of testing strategy to assess whether the E&M biotransformation really has an effect on the toxicity kind of outcome. In addition, we actually found that there was little to no difference in fixed DNA damage observed between repeated and bolus long-term exposure regimes. Now, we're currently doing a little bit further investigation, as I say, with the pro-inflammatory markers and liver functionality, but at this stage, there doesn't seem to be much difference between whether you do a bolus or a fractionated dosing regime. But yes, I'm sure more information of this will be published in due course. So if you'd like to find out anything more about the project, the links to our website and social media platforms are listed here. Otherwise, I thank you all for listening, and I hope that you found that useful in some way, and I am happy to take any questions.
Thanks. So yeah, thank you for giving me the opportunity to present here in this session. So my name is Marije Niemeijer from Leiden University, and I'm also working within the patrols. So today I will present a 3D Hep2 stress response reporter, which is imaging based to enable nanoparticle toxicity screening. So when a chemical or nanoparticle enters the liver and reaches the hepatocyte, it provokes a certain molecular initiation event. And this may lead to the accumulation of certain types of stress inside these hepatocytes, such as endoplasmic reticulum stress response, DNA damage signaling, or the oxidative stress. And a cell tries to cope with these different kind of stresses to activate specific adaptive stress response pathways, such as the unfolded protein response, oxidative stress response, or the p53-mediated DNA damage signaling. And this to try to restore homeostasis during stress conditions by these nanoparticles inside the cell. However, when the stress is too severe, a certain breaking point will be reached, and the cell switches to apoptotic signaling, which may in the end lead to either necrosis, apoptosis, and leading to certain adverse events inside the liver. So to be able to have a prediction of the development of these adverse events in the liver, it is of value to map the activation of these adaptive stress responses to allow mapping of different kind of stresses provoked by these nanoparticles at an early stage during development to this toxicity and adverse events. So yeah, a cell has different adaptive stress responses to cope with these different types of stresses. And they are regulated by a certain sensor, which controls a specific activity of transcription factors of these 4 different stress responses, which leads to the activation of certain downstream target genes, enabling the cell to remove this type of stress in the cell and to restore homeostasis. So if you can map the activation of these different pathways by looking at these specific genes at different levels within these pathways, we can have more insight of how these nanoparticles induce stress inside these hepatocytes. So to be able to follow these activation of these genes of these different adaptive stress responses, at Leiden we developed an Hep2 reporter platform by using the Bach recombinering technique. So here on the left, you see the Hep2 cell being transfected with certain plasmids containing the gene of interest with the GFP tag. And they're being transfected and selected so that we end up with cells that specifically express genes that are connected to a GFP tag. So then when a certain stress is inside the cells, we see activation and induction of this GFP fluorophore. And this we can do for all different kinds of stress responses, such as oxidative stress, the UPR, or inflammation signaling, DNA damage signaling, or other types of stresses to build up this reporter panel to follow different types of stress upon nanoparticle exposure. So here's an example how this may look like. So you have your Hep2 cells, and they're exposed to a certain chemical or nanoparticle, and then over time you can follow the induction of this GFP reporter connected to a certain key gene within these adaptive stress responses over time, and this indicates that there's a certain type of stress inside these cells. And this GFP signal you can then quantify. So here you have your image with the nuclei in blue with the GFP. You can first quantify the single cells using specific software, such as Cell Profiler. So you can define single cells based on the Hughes signal and also the complete cells. So then you can also measure the GFP intensity within the nuclei, but also within the cytoplasm of each single cell. And this you can further analyze to see, okay, which kind of nanoparticles or chemicals induce which type of stress responses at a certain concentration. So for instance, if we zoom into one of these stress response pathways, such as the oxidative stress pathway, normally NRF2 is being kept inactive by the sensor KEAP1 and is being ubiquitinated and degraded by the proteasome and is then in an inactive state when there's no oxidative stress. But when there is oxidative stress, such as by this known compound or a nanoparticle, then NRF2 is able to move away from the KEAP1 sensor and can accumulate inside the nuclei and then activates its downstream target genes, such as sulfurdoxin-1, to eliminate the oxidative stress. So here you see an example of these different reporters for each level. So we have a reporter for KEAP1, the sensor, NRF2, the transcription factor of this pathway, and sulfurdoxin-1, which is a downstream target. And you can see that we can then follow the activation of these genes over time when they're exposed to a known oxidative stress inducer. You see clear upregulation of this GFP signal. And this you can then quantify and follow the dynamics, activation dynamics over time. So here on the x-axis you see the time versus the GFP intensity, which is quantified. You see that there's a time-dependent induction and that also the different genes within the pathway have different time dynamics. So NRF2 is activated really early, followed by the activation of sulfurdoxin and also being concentration dependent. So in this way, you can follow a whole pathway upon chemical or nanoparticle exposure. So a typical chemical or nanoparticle screening setup is first seeding the Hep2 reported cell lines which are interested in a 384 well format, and then perform Hughes staining to visualize the nuclei, followed by drug exposure or nanoparticle exposure, and then do confocal microscopy to follow the activation over time. So we can do lifestyle imaging, where the cells, the plates are kept at 37 degrees, and we can image each single well every hour for instance, 24 hours or even up to 72 hours in this 2D setup. So with this data, you can extract different features. So you can extract different time responses, but also concentration-dependent response. And based on this, you can define, for instance, the point of departure, so the concentration at which we start seeing an induction of a certain stress response pathway, but also the EC50 or the slope or the maximum induction of this GFP response. And we can do this at a single cell level. But for nanoparticles, it's essential to also test them for long-term repeated exposure scenarios. So we wanted to go from 2D to 3D for this by using a spheroid system for the Hep2 cells. So here we also use 384 well plates and add 11 microliters of Matrigel, which is still in a liquid phase, so kept at 4 degrees, so that you're able to pipette the Matrigel in each well. And then these plates, which contain only Matrigel in each well, are then incubated for exactly 43 minutes at 37 degrees in the incubator. And this allows to solidify the Matrigel for somewhat. And then after this 43 minutes, 1,000 cells per well are added, which was contained in 50 microliters of medium. And this allows to that these 1,000 cells sink into the Matrigel layer. Because they are already 43 minutes incubated at 37 degrees, they will sink into the middle of the Matrigel, so not directly to the bottom, but also not on top. So with this 43 minutes, they will end up approximately in the middle of the Matrigel layer. And then after plating, the medium is being refreshed for 3 to 4 days, for at least 21 days. And this enables to let them form spheroids within this Matrigel layer. So this is how it looks like. So first on day 1, they're still single cells. So that's essential that they are single cells, because if you have already some clumps in, then they will form bigger spheroids in the end. So it's essential to have single cells to remain consistent in the spheroid size later on. And then after 21 days, you can see that they form nice spheroids within each well. And these spheroids are stable for at least 14 days, so this enables repeated exposure scenarios with nanoparticles. So we also looked at the proliferation of these Hep2 spheroids. So normally in 2-day, Hep2 cells are highly proliferative, but when they are cultured in this Matrigel layer, they stop proliferating after 14 days. Which is shown by the Ki67 staining, also seen on the right here with the IF staining. And we also looked at the differentiation status of this 3D Hep2 spheroids. We could see that they show an increased expression of albumin and also secretion of albumin, indicating that they have an enhanced differentiation state compared to its 2D state of these Hep2 cells. So we can also use this setup, so this 3D setup, in combination with this GFP reporter system to follow the different stress response pathways also in this 3D setup. So we can look at, for instance, oxidative stress using the sulfur-redoxin reporter. So upon chemical exposure, in this case, you can see a clear induction of this reporter upon exposure. But we can also do this for endoplasmic reticulum stress with 2 different reporters, but also for DNA damage signaling, where you can also see a significant increase in the GFP signal upon exposure to known stressors. So what would be the typical setup for screening of nanoparticles using this 3D setup with these Hep2 GFP reporters? So on day 0, we can seed these specific reporter cell lines in this Matrigel layer, and then refresh 2 times a week for 3 weeks to allow for spheroid formation, so they become like this in the photo on the bottom. And then we can do a nanoparticle exposure for up until 14 days repeatedly. So on day 0, we remove then the medium that's on top of the Matrigel, and then add fresh 25 microliters of medium, and then add on top 25 microliters of 2 times concentrated nanoparticle solution. And this we can do every day up until 14 days. And in the last day, we add also Hoeg staining to visualize the nuclei within the spheroids. And then after a certain amount of exposure time, we can do also, again, imaging of these 3D spheroids using confocal microscope. And then we make different Z stacks at different heights within the matrigel layer to visualize the complete spheroids at different heights. So we use here steps of 30 micrometers, also being visualized here above on the right. So then you have all these images on the different Z stacks, and then we will make a 2D projection based on the maximum pixel intensity of these different Z stacks, so that you have actually then a 2D projection of the different heights that you have imaged within each bell. And this you can then segment based on the Hughes staining using the NIST elements analysis software, and then quantify the GFP intensity within these segmented spheroids. So this is how it looks like when you would expose certain nanoparticles within these spheroids. So for instance, we have tested reporters for the UPR, DNA damage and oxidative stress. And you can see that after 14 days of repeated exposure that we see an enhanced signal of these various reporters, so that you can visualize, okay, which stress responses are being activated upon exposure to these nanoparticles. And this you can then quantify and visualize within overview graphs. So you can see that there's a dose-dependent induction of these stress response reporters for the different nanoparticles, and that it's also time-dependent. So here on the x-axis you see the different concentrations, and on the y-axis the GFP intensity that has been quantified for the 4 different reporters in this case. And you see that the 4-day repeated, so in the dark blue, you can see that at lower concentrations the reporters are already being activated, while the 5-day repeated exposure scenario, you see that only at the highest 2 concentrations we see an activation of these reporters. So with this reporter panel, both in 2D and 3D, we can do high throughput mechanism-based toxicity screening, and it enables both single and long-term repeated exposure screening in 2D and 3D setting. So as a next step, we also want to test these reporters in the hanging drop method that Sam just presented, and that we can also compare this with each other with the different setups. So hereby I would like to thank my colleagues at Leiden University, but also collaborators also in Patrols, and also for your attention. And I'm happy to answer any questions. Yeah, that's indeed a good question. So yeah, we have tested up until 100 micrograms per milliliter, and there is indeed with the highest concentrations that there's so much saturation of this nanomaterials that it will interfere with your GFP signal. But it was primarily for the highest concentration that we tested. I see another question in the chat, like about how to quantify the amount of particles within the spheroid. So that's a good question, and we are currently testing it with electron microscopy to quantify and see, okay, also where the nanoparticles end up, if only the outer cells take up these nanoparticles, and how the distribution is within these spheroids, especially since it's embedded in matrigel. So matrigel might interfere with the take-up of these nanoparticles, but still also visually we see that a lot of nanoparticles also end up on the bottom. So yeah, there is nanoparticles going through, but we still need to quantify this further. Yeah, so currently these nanoparticles that I've been testing are not tagged. So that would also be a solution to also test nanoparticles that are tagged to see what is the distribution of these nanoparticles within the spheroids. But it might maybe differ somehow a bit to the nanoparticles that we have been testing. But still, it will give a good indication of how the distribution is of these nanoparticles. Yeah, I see a question about the responses to nanomaterial exposures. But I can also definitely share the slide with you, Trine. Yeah, so within Patrols we have also work packages that focus on in vivo comparisons. So we are also planning to integrate to do the in vitro, in vivo comparison to see how it matches with each other indeed. Yeah, that's also why it's relevant to have the internal exposure of these nanoparticles to also compare it.
How about availability of such reporter cell lines to other laboratories you provide?
Yeah, so recently it's being commercialized with a company named Toxis in the Netherlands. So yeah, just recently they fused with another company, Toxis.
And are you planning to extend to further genes of interest in the reporter lines, or?
Yeah, so also based on transcriptomics data, because the initial reporter panel was based on literature, but now we are also selecting genes that are being co-regulated based on T.G. Gates dataset. So that's a chemical-based transcriptomics dataset to see which genes are now most relevant in liver systems such as the primary human PET cells and HepG2 cells, and then compare to in vivo, and which are being which are relevant in vitro and in vivo situation.